much. It was really interesting, the 3 talks. I have questions for each of you. I mean, the first one is more on the first 2 talks. And I wanted to ask you guys, you showed very nicely that at least 30%, around 30% of patients, they do present with spike and wave activation during sleep, which is actually what we see in patients. But what I don't know, because I don't know how much data we do have around, is when does this appear over the time, over the course of the disease, and if together with the appearance of spike and wave activation, you have a worsening of, let's say, non-seizure symptoms. That's my first point. And maybe I'll ask you a question later. Would you like me to go now? Okay.
Yeah, thanks a lot for this question, because we just don't know. And that's exactly why we need longitudinal data without any assumption, just look at it. And that's what we need to gather, I think. Yeah.
Okay. And then I have another question for you. I was thinking about study on EEG, which is actually a nice study, but my questions are the following. So, firstly, you're dealing with patients, the majority of them with their mental issues. So how can you be sure about the resting state? Firstly. Second question is, you at least you showed data, overall data. I mean, all data put together. So, but we actually know very well that the delta power is significantly changing over time. So if you compare a patient 12 or 18 months with a patient 10 months, it's completely different. And on top of these, the majority of those patients, they're taking anti-seizure medications or different drugs like GABAergic drugs, like benzos, and we know exactly that benzos, they may increase beta activity. So how do you deal with all of those, let's say, confounding factors? Thank you very much.
Great questions. So I would say to start that we built the paradigms to all work with all kids. So we don't necessarily only have kids with intellectual disability, and we have a lot of cognitive data from pre-COVID cohorts. And so we always look to see, do things like IQ score or Vineland adaptive abilities, are any of those modulating the signals that we see? And we haven't necessarily seen anything that is specifically driven by intellectual disability. So a lot of our measures we are hoping are going to be more impervious to that, but the important thing is to measure it and to test it to make sure that it's not just a cognition thing. Is it a GRIN thing? Is it an autism thing? We're trying to understand all of that in a very multifactorial way. So we recognize it as a compound problem. And then the other question was related to... It was on benzos medication. Right.
May change the beta power, increasing the beta, and so on.
Yes. So in the resting state data that I showed, we controlled for age. So the offset effect that I mentioned was controlling for age. And so and we do have kids, I think, in that sample from age 4 to 25, but we did not see that offset was varied by age. And so we are fairly confident in that condition effect for that piece, but that's something that we consider, and why we do a lot of longitudinal work is to really understand what is stable over time in the same way that we mentioned. And so I think that for us it's really about increasing that sample size as best as we can. And the other piece I'll mention is because a lot of my colleagues in the States don't like the fact that we come to hotels and set up. It's not as scientifically rigorous in their eyes. So we spend a lot of time in the post hoc in the way that we analyze the data. So instead of just saying that everyone's P3 is higher, we're really specifically looking at that child's condition effect, which should be impervious and consistent to what they experience in the moment. So we try to kind of take some of these behind the scenes analytic strategies to better understand across different contexts, whether or not they were in the lab or in a hotel or in home.
I really appreciated all the talks. I have a question concerning bridge between the basic and clinicians. I wonder, the development, expression of subunits encoding for glutamate receptors like 2B and 2A is obvious that it's developmental regulated, and I would expect that also the symptoms associated with mutations will change during the age. Is this the case what you see clinically? Like if you have 2B mutations, that it will be improved or vanish in time, while 2A will get worsened with age?
Thank you for an excellent question. I think, yes, we expect that. I can maybe relate to that, like our speech-language therapist already looked at our cohort of, I think, 80, which she analyzed from young children to a little bit of teenagers and an older group, and we do see that only looking at speech-language, the phenotype is changing. The thing is, yeah, we need, again, bigger N's to really see the patterns in it. So yes, and I think you see it across all the domains, right? You look at the motor phenotype, the cognitive phenotype, the adaptive phenotype, yes. And I very much appreciate saying that the fundamental scientists like you and our teams, I think it would be amazing if we can use these conferences to align some of the data, right? That we can maybe make a framework on things we measure in the patients and see, like the Drosophila work or the mice work that we are doing in our institution, how we can maybe come to these cross-species assays so we can speed up the science. Yeah.
One other thing that I was thinking about in the first talk is the fact that you're collecting the intervention data too. So we don't let our kids just live their life and wait until we understand how to solve it in the animal. We're constantly trying different things. And so I actually wonder if you could speak to any of the kind of treatment things that you have seen in your registry or data set.
Thank you. Honestly, the problem with my actual study is that it's still ongoing. So it's really hard to have a conclusion for now about this. But what you were thinking just before is that really with time things are going better, especially on the speech. And it's hard to say if it's the treatment or if it's just ongoing of the disease. So.
Thank you. I'm Jean Thomas. I'm a parent, not a scientist. My son has GRIN 2B, and he was born in the US, and I'm French, and we live in Belgium, so I feel connected to all of you. And I will come to talk with you at the break. I was very interested on the autonomous system marker, because indeed when, in the things we notice for our son is, and it's written sleep disorder, he only wants to sleep with light on, which is curtains open. Also temperature body, I think, disorder, because he sleeps summer with winter pajamas, winter pajamas and never a blanket on. Some things that are very different than I would think what we would normally do for sleeping. But my question was also, he had, so he was diagnosed relatively late, and he had, so we started with EEG, why he doesn't speak, he had speech delay, was the main. And so he was treated with Depakine, and eventually we stopped the Depakine, and the EEG became normal. So, or at least I was told it's normal. So I was, indeed, it was touched, when does it appear, but there is also when it disappears question, because it seems in our case that it disappeared, and that's what we had been told, that it could evolve like that. So, which I thought.
Yeah, it works. Well, thank you. This is very, it's a good observation. It might have been gone away while you were treating, but it can also be an effect of the Depakine itself, to be really honest. We know that you can get encephalopathic, of using Depakine. So when the switch was, we don't know in retrospect. So yeah, I think we need to, in one way or another, we need to be able to predict or to find a measurement to measure within treating, are we there or are we not? But we can't do that at this moment. No.
And I think to comment on your sleep disturbance, I cannot show you the data yet, but we are looking into that, because I think all the GRIN parents that come to us tell us there are sleep problems. And we feel like just pattern recognition, you know, as a physician, like most of the kids improve around pre-puberty, we feel. I would like to measure that again also, right? So probably this is how I view it, like also maybe a developmental delay in getting your normal sleep patterns. And that is part of the GRIN disorder. I think it would be very interesting also with animal models like the Drosophila, because you can really study that well, look at active and sleep patterns in that. There's much work to do.
But there seems to be hope in terms of sleep as we go on.
Yes, that's always what I say, because we as physicians feel as frustrated as you, because we have melatonin and a few other things, and then we send you home with your kids that don't sleep. And we are very appreciative of that.
There's one in the back. Should I go now? Okay. Hello. Thank you all for the amazing presentations. On behalf of my GRIA-1 child, I would like to thank you for the excellent work you're doing, and it really starts to show where the science of it is actually coming into practice. On that, we see the importance, I think, across all the presentations of EEG collecting data and clinically, but also then analyzing it in studies. And I guess I was a bit shocked in a very positive way about this beautiful new EEG that there was even videos of, and I was just wondering when we could have that. But the question would be more, is there a difference in the data obtained across the different types of EEGs? Because we've been doing sleep deprivation and 24-hour video EEGs. So what is the quality? What do they tell you if you compare them across different types? And then on the importance of the data itself, can we also have, as we have other registries, should we have an EEG registry? I'd be happy to provide all of mine in case that's important to the research. Thank you.
To start with the first, we should have an EEG registry, I think, because with all the new techniques, it's pretty easy to combine things, and so yes. But you're very right, you can only do this type of EEG for a short period of time, because it comes with wettening the cap, and once it dries, you will lose the signal. So you can't do 24 hours, you can't do sleep deprivation and wait until they sleep, and it will loosen in that time. So yes, the quality of a normal adjusted EEG is higher in that sense, and it tells you a lot more. So yeah, we also need to work on those techniques, I think, but that's for the broad population with epilepsy. Yeah.
So the other thing I just want to add is that I think that the push needs to be to combine, like mine is entirely a research space because we're doing these tasks. Again, it's just 20 minutes. It's not meant for long-term recording, but what we learn from that could be things that we could also pull out of clinical EEGs. So kind of using a collective and thinking through how we can kind of combine efforts. And there's a paper in another group I've worked with where they did use the clinical EEGs to really understand and decode what they call microstates. So trying to understand kind of common patterns in which these kids' brains were in different states of being. And I think that that's something that we could do both on the research and clinical side. So it starts with creating a registry and then sharing data internationally and growing that. And there's a bit of technical hurdles, but I have ideas if anyone has the money.
Related to the exchange of data and collecting data, Corinne, what are your intentions with opening the doors to beyond France, across Europe and further? What's in the works there?
So my work is only on France, and for now, but of course when we have the data afterward, we manage to share it with the people that want to start studies or anything else. And for the moment, really for regulatory reasons, I only stand to France. Even Belgium or Switzerland, we meet from parents yesterday. That could be included, but for now, really for a mandatory reason, it's a little bit hard to go out of the frontier.
Okay, I think that's something we as an association should start putting on the agenda to help facilitating and to make sure you have at least pan-European, IRBs and GDPRs and all that in place. Because I see a tremendous potential in getting natural history collected centrally from different partners. Now, where it is at the end, it doesn't matter, but it needs to all come together at some place.
Hi, I have a question about if we have any information about a biobank of maybe blood, CSF, urine maybe, with the gene expression analysis in one of those registries, so we can have more information about what's going on, and which is not in an EEG setup.
Yes, when we started, I would have wanted to include already the informed consent for the biobank, but you can only do a few things at once. So I think, we have a small biobank for GRIN 1 variants for IPC. So I think if I look to my team, we are more than willing to share this with the scientific community. We are working hospital-wide on a biobank, which means that if you enter our hospital, and usually via the genetics department, we are building that. So at now, I would think probably for many of the subjects, we would have blood from metabolic screen, which we may, if we can work it out, look at, because that's stored as well. But of course, it's something definitely we should look into also, yeah.
I'll just say that I collect data at the same time as Simon's Foundation and Combined Brain. So we have memorandums of understanding in place to be able to share data across platforms. So they collect the biospecimens. So if you see like an iPSC line, for example, in Simon Safari, their iPSC lines, it's likely that I've seen one of those kids. So there's potential opportunities, and we're still kind of growing how to make that more clear. And they don't have our raw EEGs yet. We're still kind of working on that. But if there's interest and you know of samples on the American side, let me know.
And I'm thinking we could share the children's variants with you. And if any of you is interested in a certain variant with certain specimens, we could do it when they come to our clinic and then send it to people, right? That's a possibility as well.
I think it was Quarantine that you mentioned, I know you don't have yet all the information about treatment, but you mentioned that the GRIN 2As were responding to glucocorticoids, right? Or
it's only on CSVS, so the...
Okay. Yeah. Okay. No, because this is something that we're wondering, especially with the spike wave, if there is an underlying, neuroimmune issue going on with these kids. But, it would be something that, we've been trying to push for, to maybe test on GRIN because we don't understand. Oh, sorry. Yeah.
Well, I think we think the same, but many of these epileptic encephalopathies like in the West crisis, like the epileptic spasm crisis, glucocorticoids work. And in ESAS or CSWS, it also works. So yes, there is an effect, but we're not sure what the effect actually is. I think this means that we should, for now, it's just meant to cover the crisis and not to have as a prolonged treatment. Also seeing the side effects. But it's a nice thing to study, really. Why does it work? Is it a direct effect of the corticosteroid itself, or is it a bigger effect? So, yeah.
Can I add something on the use of steroids? Yeah, which is something that is actually not specific to the 2A, GRIN 2A. I mean, we use steroids in definitely almost all developmental and epileptic encephalopathies in which we do have spike and wave activation during sleep. And when spike and wave activation is strictly related to a worsening of cognitive condition, okay? And unfortunately, it is working, but the effect is really temporary. And let me say, it is 30% of patients, 40% of patients with DEEs improve temporarily with steroids. So, guys, yeah, no, because there are families here. No, yeah, that's why I want it really to be really super transparent, and everyone should know. And there is a nice study which was published 2024, I think, from Flor Jansen. You know the study. And on spike and wave activation during sleep. And definitely steroids are a little bit better than benzos, but results are not really exciting. Still we use it because we have a certain percentage of patients which improve. So I'm not saying not use, but yeah, thank you. Oh.
I completely agree with Nicola about the use of steroids, that we still, even if it is not very much effective, not so much as we would wish, but still we can use it with this about 40% of patients effective. But maybe, at least in tuberous sclerosis, we used, we had also intramuscular corticosteroids before. Now we have only this probably oral. But in these children, when they had the beginning of epilepsy in the first year of life, it was even, according to my opinion, it's even, if you are using and you can get this temporary effect, you may just preserve the cognition for this particularly sensitive period of brain development. So later on, it's not so, effective. But for these children below 2 years of age, it's very, very important.
Since we see, always tremendous effects giving a little bit of ibuprofen for a few days, in our dog, since years, the families always comment, our kid gets out of a short infection with flu better than she was before. And in our case, just 2 months ago, the clonal seizure stopped. 5 days of ibuprofen for an influenza A infection. So, quite amazing. In that regard, have people, and since NMDA is a target of autoimmune encephalitis, inflammation, have people actually looked at an autoantibody against NMDA in the mutated context? Is that being done? We're talking to Josep Dalmau in Barcelona about this, to really clone those things and test whether there is an underlying autoimmune component to that will contribute to inflammation.
Regarding ibuprofen, you have a lot of confounding factors. Maybe it's the infection and the rise in natural steroids that works. Maybe it is the not eating, so it is a ketosis state.
I fully agree with you, and that's the neurologist. Angels was the first thing that she said, well, yeah, you see that with infections, even if you were not to treat with ibuprofen. But we also see improvements if we treat with ibuprofen, and we don't do this daily. We are conscious of the liver health, at least in the daughter. But we don't, for pain, for instance, menstrual pain or so, and you sometimes also see there an effect. I don't want to oversell it. It's just because so many families have seen this and commented for years about that. I think we shouldn't ignore immune inflammation.
Or just neuroimmune markers, biomarkers, that maybe we can pinpoint, that there is something that could be targeted, that it doesn't need to be a glucocorticoid, because we know in most diseases, glucocorticoids stop working anyway.
Which at least in CSF, in a group of about 20 or so GRIN kids, was not really elevated in the CSF pro-inflammatory markers, but it was compared to a group of autistic kids, so I'm not sure the control group was the right thing to do.
Maybe in that line of thinking that, we know of a few GRIN subjects who a little bit later in life developed chronic, inflammation of the bowel. That it was with very a-specific biomarkers called protactin and all that, but that's also something that we're I think it's interesting to pursue, right? Whether it's in a certain subgroup, whether this is an autoimmune effect or a secondary effect because of their intestinal issues, motility issues, we don't know, but I think it's, there are some cues there, I think.
Do you look into, sorry, I don't want to get carried away too much in autoimmunity, but do you look into HLA polymorphisms in those kids? I mean, are they all predisposed to mount the ones that you say that will later on develop these IBDs or so?
No.
You may have the data from the genetics.
Good. Any more questions? No?